In this video, we will discuss about the role of glycan in the leukocyte capture. These leukocytes are the white blood cells, and it has an important role during the immune response. So we will understand how these leukocytes get attached to the endothelial cells, and what are the ways and what are the changes in the glycosylation that might affect this leukocyte capture. So we will start with this normal conditions, that is on the left hand side, where the leukocytes has different kind of we can say cell surface protein, such as this CD44, and so, and at the same time these endothelial cells will also has different cell surface proteins such as selectin, hyaluronic acid. So what will happens in this case when the this galactin 1 interact with this leukocytes, then it will inhibit the leukocytes capture to the endothelial cells, whereas galactin 3 will support the leukocytes captures to the endothelial cells. And at the same time there is this hyaluronic acid. When interacts with these leukocytes, it helps the leukocytes for it helps the leukocyte to capture uh, or it reduces the rolling speed of the leukocytes and helps in the capture of these leukocytes to the endothelial cells. And whereas when this CD44 which is over which interact with the hyaluronic acid, at the same time the ligands that are expressed on the leukocytes also interact with these selectins based on the different carbohydrate structures, and those carbohydrate structures are shown here, where we can see that the SLEX, this kind of structures, Silai-Lewis X structures that are expressed on the leukocytes, are more prone to interact with these endothelial cells, and thereby they help in this leukocytes adhesions to the endothelial cells. So, and these Silai-Lewis X structures are also found in higher quantity or higher expression on the cancerous cells. Thereby, this kind of structures helps on the cancerous cells, helps them to metastasis at the secondary organs. So this is about the normal process. Now let us understand when what will happen when there are some changes in the carbohydrate structures observed. So when this TNF alpha is upregulated in this endothelial cells, it will lead to the decrease in the mannose, and decrease in the mannose will cause upregulations of the mannose, and that will lead to the high end glycans on this ICAM1, and this high end glycans on this ICAM1 will promote or accelerate the CD61 plus monocytes attachment. And so this upregulations of this N-glycans on the ICAM will enhance CD66 monocytes attachment to the endothelial cells. And in other case, what will happen is that when this CCL21 interacts with this heparan sulfate that is shown here, it will promote the T cells adhesions to the endothelial cells. And these kind of adhesions are required whenever there is a requirement of the immune cells at a specific sites. So these kind of combinations might occur at the endothelial cells. In other case, what will happen when the silated CCR7 are expressed on the endothelial cells, then they will prevent the binding of neutrophils or the adhesion of the neutrophils and the monocytes. And these things are usually happens when there is when the requirement of the immune cells is reduced at the site of infection, or there is no requirement of the immune cells at a particular site. And in the last case, what will happen is that when monocytes are stimulated with INF alpha, that will lead to this NACT enzymes degradations, and it will cause the decrease in the glycosylation. In more specifically, the beta 1,6 glycnec will decrease on the alpha 5 and beta 1 integrin, and this will cause the high upregulations of this FAK and leads to the high upregulations of this ERK kinase, and that will enhance this monocytes adhesion. That will enhance this monocyte adhesions to the endothelial cells. So in this case, the interferon gamma enhances the monocyte adhesions, whereas the silated CCR5 expressed on the not silated CCR7, sorry, I want to correct one thing. Silated CCR7 are expressed on the T cells, and they can interact with the CCL21 and heparin that are expressed on the endothelial cells, and these combinations helps in the adhesion of the T cells. Whereas when the galactine-3 and CXCL12 form a complex in the endothelial cells, they will prevent neutrophil and monocytes adhesion to the endothelial cells. So this is the corrections that I want to make. In previously I mentioned or wrong function about the silated CCR7. So this neutrophils and monocytes adhesions is inhibited by galactine-3 and CXCL12 complex, and thus whereas this interferon gamma stimulations on the monocytes will enhance the monocyte adhesions to these endothelial cells. So this is just a overview of how these glycans can affect the leukocyte capture on the endothelial cells, and this property of the glycans sometimes is being exploited by the cancerous cells for the metastasis or migration. So I hope you got a basic idea about the role of glycans in the leukocyte capture. If you need to understand more about this glycans in the roles of glycans in the leukocyte adhesions, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.